In part A of the lab, we're going to set up 8 test tubes and label them A, B, C, D, etc. A will be fructose, B is glucose, C is lactose, D is sucrose, E is xylose, F is our unknown, G is gelatin, and H is honey. Now we're going to add about 3 milliliters of each solution to the appropriate test tube. So for example, we'll start with fructose, test tube A, and we're going to use a disposable pipette to add about 3 milliliters of fructose to test tube A. So I'm just using the lines on the disposable pipette to estimate 1 milliliter, and then I'll do that 3 times. Now we'll move on to glucose and do the same thing, and we'll just keep going down the line. Once we've added 3 milliliters of each solution to the appropriate test tubes, let's just take note of their colors or lack of color. Most of them are colorless. Gelatin has a bit of a yellowish green color, and honey is a little bit orange. It's very dilute though, so it's not the pure orange that honey normally is. Now notice that the unknown is also colorless, so that could help us determine how it's the same or different from other solutions. Now we're going to add a 3% yeast solution to each of these test tubes, and remember that yeast contains the enzymes needed for fermentation. So again, we're going to use a disposable pipette to add about 3 milliliters of the yeast solution to each test tube. So notice that fructose initially was colorless, but once we added the yeast, it turned the same color as the yeast solution. And here you can see all of the test tubes with the sugar and yeast solution. They all look pretty similar. We're just going to mix them and then add them to a hot water bath. Earlier, I set up a hot plate with a beaker that was about 2/3 full of water. I turned on the heat setting, and then I also have a thermometer attached with a thermometer clamp to a ring stand because we need the water to be about 45 to 50 degrees Celsius. So once the water is heated sufficiently, we are going to add our test tubes with the sugar solutions and yeast solutions to our hot water bath. What we're looking for here are the formation of bubbles and really just any changes from the original appearance of the solutions. So here you can see some of the test tubes have already started producing a gas, which is CO2. So there's some bubbles forming there. Once we've let them sit in this hot water bath for a few minutes, about 30 to 45 minutes, then we're going to check each test tube and make some observations. After 30 to 45 minutes, we can now check our samples for the formation of CO2 gas, as well as any foam that might have formed. So let's look at our first test tube A, which is fructose. There was definitely a foam that formed at the top, and we can also see some bubbles coming up from the bottom. It might be hard to see on the screen though. Test tube B was glucose, and there's also a foam that formed at the top. So that definitely tells us that we had some CO2 bubbles that were also forming during this process. Test tube C contained lactose. You can see bubbles are still forming inside the test tube. So that definitely produced CO2, although there isn't really a foam at the top like we saw in A and B. In test tube D, we had sucrose. There's a little bit of foam at the top, and there was definitely some bubbles within this test tube. It might be kind of hard to see though on the screen. In test tube E, we had xylose, and you can see bubbles are still forming within the test tube, so there was definitely carbon dioxide gas forming. Test tube F was our unknown, and you can see bubbles coming up from the bottom. So CO2 definitely formed with our unknown, although there wasn't a lot of foam at the top. Test tube G was gelatin, and gelatin did have carbon dioxide gas bubbles coming up from the bottom. And finally, test tube H was our honey sample, and that also had some bubbles forming, so it did have some carbon dioxide gas. Although again, it might be hard to see on the screen. In part B of the lab, we're going to set up again 8 test tubes with their labels, and we're going to use the same labels as before. So we're going to add 20 drops of each sugar solution to each appropriate test tube. So instead of the milliliter amount, this time we're counting drops. Once we've added 20 drops of each sugar solution to each test tube, we're going to now add 20 drops of Benedict's reagent or Benedict's solution to each test tube as well. Here you can see the color of the Benedict's reagent is a light blue. So once we add 20 drops of Benedict's reagent to each test tube, those solutions will also turn blue. Here you can see all of the samples are blue, similar to the Benedict's reagent. Now we're going to add these to a hot water bath and see if those colors remain or if they change. This time we want our hot water bath to be boiling instead of at a particular temperature, so I removed the thermometer. And I also added 2 to 3 boiling chips so that the water wouldn't boil too much or overflow. So now I'm adding the test tubes with our different sugar solutions and the Benedict's reagent. And I'm going to speed up the video so you can watch the colors change. After a few minutes, we're going to remove the test tubes from the hot water bath. What I'll do here is just use some beaker tongs to remove the beaker of test tubes and let them cool a little bit, and then we'll observe the color changes. Remember that Benedict's reagent will oxidize reducing sugars. So if you see a color change, that means oxidation occurred. So here's fructose in test tube A, and feel free to pause the video to make your observations. Here's test tube B, which was glucose. And if you're having trouble distinguishing between these different colors, let your instructor know. Here's test tube C, which was lactose. Here's test tube D, which was sucrose, and it looks like it remained that same blue color as Benedict's reagent, so that tells me a reaction did not occur. Here's E, which was xylose. And here's F, our unknown. And note if it looks similar to any of the other sugars that we saw. Here's test tube G, which was gelatin. Again, this remained a clear blue color, so likely no reaction occurred. And here's test tube H for honey. Benedict's reagent does contain copper ions, and this could harm marine life if we put it down the sink. It could also affect our pipes, etc. So we're going to pour these solutions into a waste container, and we'll handle that later on. So here I'm just going to pour the contents of each test tube into our waste container, which is appropriately labeled with the name of the lab and what the main component is that we're trying to collect, which is Benedict's solution. Now I'm going to rinse the test tubes a little bit with water just to get as much of the solution as possible out of the test tube. It's okay if a little bit remains in the test tube. We can wash that with soap and water down the sink. In part C of the lab, I already added the appropriate amounts of each sugar solution to the appropriate test tubes. Now we're going to add a reagent called Barford's reagent, and we're going to add about 20 drops of this reagent to each test tube. Notice that the color of Barford's reagent is really similar to Benedict's reagent. That's because they both contain copper, and in certain forms, copper is a blue color. So that means that this is also an oxidizing reagent, similar to Benedict's reagent. Now we're going to slightly mix each test tube, and then once everything's mixed, we're going to add these to a hot water bath again. So here's our boiling water bath again, and we're just going to place our test tubes in here and watch the color changes. Now, Benedict's reagent was capable of oxidizing both monosaccharides and disaccharides, but with Barford's reagent that we're using here, it's only capable of oxidizing monosaccharides. So we might notice some differences between the last test and this one. Now we'll remove the beaker from the hot plate using our beaker tongs again. Then we'll observe any of our reactions. So let's observe the color changes that occurred for our different test tubes, if any. So here's test tube A, which again is fructose. Notice that parts of the test tube are a reddish color, but parts still look sort of blue. Test tube B is glucose, and once we shake the test tube, it starts to turn red, but it does still look like there's a little bit of blue in there still as well. C is lactose. Notice that there's no color change. It's still the blue color of the Barfid's reagent. Same with sucrose. There was not a color change. E was xylose, and again there's a bit of red in there and a bit of blue that remains. Our unknown did not change color. It stayed the same blue color as the Barfid's reagent, so there was no reaction. Gelatin is also blue, but it's a little cloudy, so maybe a slight reaction occurred. And now for honey. Honey is also partly red and partly blue. Now we do want to collect the waste into a separate container here because Barfid's reagent does contain copper ions as well. But we're going to collect the Barfid's reagent solution separately from the Benedict's reagent solutions. Now notice that in some of these test tubes we do have a precipitate that's at the bottom. So I'm just going to add a little bit of deionized water and just make sure that I get as much of that solution into the waste container as possible. And I'll do that for all of the other samples as well. In part D of the lab, we're actually going to start by adding something called Seliwanoff's reagent into each test tube first. So we're going to add about 2 milliliters into each test tube. After we've added this reagent to each test tube, we're then going to add just 1 or 2 drops of each sugar solution to the appropriate test tubes. Now we're going to mix each of the test tubes to make sure everything is combined properly. Then we're going to place each of the test tubes into a boiling water bath, similar to our previous tests. Now this test is going to help us identify if we have a ketohexose, which is a 6-carbon chain that has a ketone attached. Now this could be for monosaccharides or disaccharides. So again, once the reactions are complete after about 3 minutes, we're then going to remove the beaker from the hot plate and let it cool. Now let's observe any color changes, and this will tell us if we have a ketohexose. So let's look at fructose. Make sure to record any color changes that you observe. Next is glucose. There does not appear to be a color change here, it's still colorless, so we'll just mark down colorless. Here is lactose. Here is sucrose. This one is xylose. Here is our unknown. Next is gelatin. And finally, our last test tube, honey. In part E of the lab, we're going to perform the aniline acetate test. This test is going to tell us whether we have a pentose or a hexose sugar. Remember, a pentose has 5 carbons in its chain, whereas a hexose has 6 carbons in its chain. So first we're going to add 2 milliliters of our xylose solution to the Erlenmeyer flask. And then I already mixed together the 10 milliliters of water and 10 milliliters of acid in a graduated cylinder. So I'm going to add that to the Erlenmeyer flask as well. Next, we're going to gently boil this solution on a hot plate for 1 minute. Once the solution is boiling, I'm going to add 3 drops of an aniline acetate solution to a piece of filter paper. So I'm doing that off screen. And then we're going to set that filter paper with the aniline acetate solution on top of the Erlenmeyer flask. Now, any fumes or gases that are coming off of our solution will react with the aniline acetate on the filter paper. If the filter paper turns bright pink, that means that we have a pentose, a 5-carbon chain. If the paper turns slightly pink, that means we have a hexose, a 6-carbon chain. So here we have a very bright pink color that formed from the reaction of the vapors with our aniline acetate solution on the filter paper. So that means that xylose contains a pentose, a 5-carbon chain. Now we're going to perform the same exact test, but this time, instead of xylose, our solution contains our unknown. So this test will tell us if our unknown is a pentose or a hexose. So again, after about a minute of boiling, we're going to add a piece of filter paper to the top of the Erlenmeyer flask, and this will have a couple drops of aniline acetate solution on it. And again, if we see a bright pink color, that means it's a pentose. If we see a very slight pink color, then it is a hexose. Okay, so this is our results for our unknown. There is some pink there, but it's not quite as bright as the results from xylose. So this would lead me to conclude that our unknown is a hexose. You can also see the color differences between our previous test with xylose and our current test with the unknown.